Hello everyone. In this video I'll be teaching you on the basic learning of CT head anatomy. The topics I'll be discussing in this video will be how to differentiate CT from MRI of the brain, how to identify different lobes of the cerebral hemisphere, how to identify gray matter and white matter in the brain, what are ventricles of the brain and how do we identify them, what are cisterns and where are they located, what are the parts of basal ganglia and internal capsule? How do we identify different parts of the brainstem and cerebellum? Welcome to Radio Clinics. How do we differentiate CT from MRI of the brain? CT scan can be differentiated from MRI of the brain by looking at the bones. Since CT scan is actually based on X-rays, which means that CT scan image forms in similar manner to the working principle of X-rays. CT scan machine is actually a large X-ray machine in itself. If you look at this image, you can see that the bones are white in the X-ray. The ribs are white, the clavicle is white, the scapula is white, but the lungs are dark. Similarly, in the CT scan, the bones are white. This is the skull bone and it appears white in the CT scan. MRI on the other hand is magnetic resonance imaging. In MRI scan, the different areas of the body give different signals depending upon the sequences. For example, in a fat sensitive sequence like a T1 weighted MRI, the bone may appear slightly bright because of the presence of bone marrow which contains fat. But in a T2 weighted MRI, which is a fluid sensitive sequence, the bone appears dark and the fluid inside the ventricles, they appear bright. But in a CT scan, the bones are very white, which helps you differentiate it from MRI scan. Furthermore, if you look at the MRI scan, you can see that in the right hand corner you will have the sequences of the MRI given. The TR, TE will be given in the MRI scan, but in the CT scan these informations will not be provided. This also helps us to differentiate CT scan from the MRI scan. How do we separate different lobes of the cerebral hemispheres? Brain consists of 4 lobes. They are the frontal lobe, parietal lobe, temporal lobe and the occipital lobe. There are 2 cerebral hemispheres each containing these 4 lobes. As in this diagrammatic illustration, you can see that the frontal lobe is separated from the parietal lobe by the central sulcus. The temporal lobe is separated from the rest of the lobes by the sylvian fissure, and the occipital lobe is separated from the rest of the lobes by the parieto-occipital sulcus. But in a CT scan, to understand the demarcation of the lobes, first you will need to have basic knowledge of the skull base anatomy. The skull base has 3 cranial fossas as shown in this image. These are the anterior cranial fossa, the middle cranial fossa and the posterior cranial fossa. The anterior cranial fossa is separated from the middle cranial fossa by the lesser wing of the sphenoid bone, and the frontal lobe is present in the anterior cranial fossa here. The middle cranial fossa is bounded anteriorly by the lesser wing of the sphenoid and posteriorly by the petrous pyramid of the temporal bone. The temporal lobe is present in the middle cranial fossa. In the posterior cranial fossa, there are the posterior fossa structures like the brainstem and the cerebellum, which is bounded anteriorly by the petrous pyramid and posteriorly by the occipital bone. So here, the anteriormost part of the cerebral hemisphere, which is bounded posteriorly by the lesser wing of sphenoid, is the frontal lobe. And here, the part of the cerebral hemisphere lying in the middle, in between the sphenoid bone anteriorly and the petrous pyramid posteriorly, is the temporal lobe. And the part of the cerebral hemisphere that lies posterior to the petrous pyramid is the posterior fossa, and it contains the cerebellum and the brainstem structures. Now as we go cranially, the parts of the skull base are not visible, So the rest of the brain is separated by different sulci. The frontal and the temporal lobes are separated by this cistern, which is known as the Sylvian cistern. So the part of the brain that lies anterior to the Sylvian cistern is the frontal lobe, and the part of the brain that lies posterior and peripheral to the Sylvian cistern is the temporal lobe. To identify the occipital lobe, you should look just posterior to the temporal lobe. This part which lies posterior to the temporal lobe and is contiguous with the temporal lobe is the occipital lobe. So this part is the temporal lobe and this part of the brain is the occipital lobe. The occipital lobe lies just cranial to the cerebellar hemispheres. So this is the part of the cerebellum. So the part that lies cranial to the cerebellum as we migrate cranially is the occipital lobe and it is contiguous with the temporal lobe. As we see in this image, as we go more cranial above the level of the ventricles, the occipital lobe is no longer seen in the sections, and the only lobes that we have here are the frontal and the parietal lobes. And furthermore, occipital lobe is separated from the parietal lobe by the parieto-occipital sulcus, and the parieto-occipital sulcus is extending posteriorly as we go cranially. In the inferior portion of the brain, we can see the parieto-occipital sulcus as a horizontal line. But as we go cranially, the parieto-occipital sulcus migrates posteriorly and it disappears at the level of the ventricles. So above the level of the ventricles, we no longer see the temporal or the occipital lobe, and the only lobes that are present here are the frontal and the parietal lobes. The frontal and parietal lobes are separated from each other by the central sulcus. The central sulcus can be identified by its omega-shaped appearance. This is the shape of omega, and if we look closely, we can see the omega-shaped sulcus right here. This is the central sulcus. So the part of the brain that lies anterior to the central sulcus is the frontal lobe, and the part that lies posterior to the central sulcus is the parietal lobe. Sometimes you might be confused about the location of the central sulcus, and you should always remember that as we go cranially, the central sulcus lies posteriorly. And if you are confused, you should note that the gyrus in front of the central sulcus is thicker than the gyrus posterior to it. That means that the precentral gyrus is thicker than the postcentral gyrus. How do we differentiate grey matter from the white matter of the brain? The grey matter of the brain consists of cell body of neurons. Hence, the grey matter is more dense and appear bright. Note that the grey matter lies in the periphery of the brain. White matter, on the other hand, is consisting of axons which are covered by myelin, and myelin contains lipid. So the white matter, because of the presence of lipid, appears slightly dark or hypodense on CT. So this part of the brain which lies in the periphery is the grey matter, while this part which is darker and lying in the central aspect of the brain is the white matter of the brain. What are ventricles and how do we identify them? Ventricles are parts of the brain that form and help in circulation of the cerebrospinal fluid. The cerebrospinal fluid circulates within the spinal cord and around the brain and helps in providing nutrition to the brain and also helps in removal of waste material. There are two lateral ventricles, one third ventricle and one fourth ventricle. So the cerebral spinal fluid is produced in the ventricles which migrate into the third ventricle and then into the fourth ventricle and then migrates around the brain and within the spinal canal and around the spinal canal. Because of the presence of fluid, the ventricles appear dark on CT scan. These are the lateral ventricles. So there are two lateral ventricles, this one and this one. This slit like hypogenicity in the center is the third ventricle of the brain. This triangular or square shaped CSF filled space lying in the posterior fossa is the fourth ventricle. The lateral ventricles join the third ventricle by the foramen of Monro shown here. So we can see these are the lateral ventricles and these are joining by the foramen of Monro, communicating with the third ventricle here. The third ventricle joins with the fourth ventricle by the aqueduct of Sylvius. So this is the aqueduct of Sylvius and we can see that this is joining to the fourth ventricle here. So this is the sagittal section of the brain and we can see that this is the lateral ventricles. This is the part of third ventricle and this ventricle, which is triangular shaped lying in the posterior fossa, is the fourth ventricle. This fourth ventricle is joining with the third ventricle by this narrow channel which is known as the aqueduct of Sylvius. What are cisterns and how do we identify them? Brain matter consists of elevations known as gyri and depressions known as sulci. So if this is the part of cerebral hemisphere, then these elevations are known as gyri and these depressions are known as sulci. In the basal region of the brain, there are deeper areas in the brain which are made by Mother Nature so that they allow for vessels to pass easily through them. These deeper areas are known as cisterns. So this area is a cistern. As we go downwards towards the base of the brain, we can see that there are multiple deeper CSF filled areas and these are known as cisterns. This cistern which separates the frontal lobe from the temporal lobe is the Sylvian cistern. So this is the Sylvian cistern. This cistern which lies above the sella turcica is the suprasellar cistern. This cistern which is lying posterior to the midbrain is the quadrigeminal plate cistern, and this cistern which lies in between the peduncles of the midbrain is the interpeduncular cistern. This cistern which lies around the pons is the peripontine cistern. And we can see that in the cisterns there run linear structures. This linear structure is the middle cerebral artery. This dot-like structure is the basilar artery. So we can appreciate the blood vessels running in the cisterns. This is why Mother Nature has given us the cisterns, so that blood vessels can run through them in an uncompromised manner, even in case of cerebral edema. These cisterns are important because in case of a spontaneous subarachnoid hemorrhage, blood accumulates in these cisterns. How do we identify internal capsule? As we can see in this image, informations are transferred from the brain to the spinal cord or from the spinal cord to the brain via channels. These channels consist of axons and these axons converge into an area which is a L-shaped structure, and this L-shaped structure is known as the internal capsule. This acts like a door which communicates in between two important rooms. So this L-shaped structure is the internal capsule. It has three main parts: the anterior limb of the internal capsule, the genu of the internal capsule, and the posterior limb of the internal capsule. Since the internal capsule is composed of white matter tracts, and white matter tracts, as we had discussed earlier, contains axons covered by myelin, so they appear dark or hypodense in a CT scan because of the presence of myelin containing fat. So in this image we can see the outline of the white matter here, and if we focus on this white matter, we can see that these white matter are now converging into a L-shaped structure here. So this L-shaped structure is the internal capsule which lies adjacent to the lateral ventricles. This is the anterior limb of the internal capsule, the genu, and the posterior limb of the internal capsule. Similarly, in the right side, this is the L-shaped structure, which is the internal capsule. How do we identify basal ganglia and thalamus? As we had already discussed earlier, most of the grey matter lie in the periphery of the brain and white matter lie in the central aspect of the brain. But in the deepest region of the brain, around the ventricles, there is a bundle of grey matter. This bundle of grey matter forms the deep grey matter nuclei and consists of the basal ganglia and the thalamus. The basal ganglia consists of lentiform nucleus and the caudate nucleus. So as we had discussed earlier, this L-shaped structure is the internal capsule, and deep grey matter nuclei, which consist of basal ganglia and thalamus, lie around the internal capsule. So this lens-shaped structure lying lateral to the internal capsule is the lentiform nucleus of the basal ganglia. The part of the basal ganglia which lies anterior to the internal capsule is the caudate nucleus, and the part of the basal ganglia which lies just lateral to the third ventricle and posterior to the internal capsule is the thalamus. We can easily remember T for thalamus, which lies adjacent to T for third ventricle. What are the structures in the posterior fossa? The structures in the posterior fossa are separated from the rest of the brain by the tentorium cerebelli. The posterior fossa structures consist of cerebellum and the brain stem. So as we can see in this image, this part, which is extension of the dura mater, is the tentorium. This is the left sided tentorium and this is the right sided tentorium. The part that lies caudal to the tentorium cerebelli are the posterior fossa structures, and it consists of the two cerebellar hemisphere. This is the right cerebellar hemisphere and this is the left cerebellar hemisphere, and part anterior to the cerebellar hemisphere are the part of the brain stem. The part of the brain stem consists of midbrain, pons and medulla. Midbrain is the cranial most part of the brain stem, pons is the mid part, and medulla is the inferior most part of the brain stem. In an axial image, midbrain can be identified by the V-shaped structure. Pons is the bulbous structure in the posterior fossa, the thickest structure of the brain stem, and medulla is the inferior most structure of the brain stem, which is thinner than the pons. We can also identify pons by looking at the fourth ventricle. So the part lying anterior to the fourth ventricle is the pons. So if we focus on this sagittal image, we can see that the supratentorial region of the brain is separated from the posterior fossae structures by the tentorium cerebelli. This tent-like structure is the tentorium cerebelli. The posterior part is the cerebellar hemisphere. The central CSF structure is the fourth ventricle. The most bulbous part is the pons. Caudal to the pons lies the medulla, and cranial to the pons lies the midbrain. So to summarize this video lecture, CT can be differentiated from MRI by looking at the bones. Bones are white in CT like in an X-ray. Cerebral hemispheres are divided into frontal, temporal, parietal and occipital lobes. These can be divided in a CT scan by looking at the bones, the fissures and the sulci. Ventricles appear hypodense on CT scan. There are two lateral ventricles, a slit-like third ventricle and a fourth ventricle which lies in the posterior fossa in between the pons and cerebellum. Cisterns are deep sulci in the brain which transfer blood vessels. Internal capsules are L-shaped structures which transmit information from brain to spinal cord and vice versa. Basal ganglia lie around the internal capsule and consist of lentiform nucleus, which is a lens-shaped structure, caudate nucleus anteriorly, and thalamus, which lies by the side of third ventricle. Posterior fossa structures consist of midbrain, which is the V-shaped structure, pons, which is the bulbous and thickest part of the brain stem, and medulla, which is the inferior-most part of the brain stem. Posterior fossa structures also consist of cerebellar hemispheres. Thank you.